The following images give an example of what transcription is. The first image being a representation of the base codes in double-stranded DNA, and the second, an example of mRNA created by the use of free nitrogenous bases to complement the antisense strand of DNA to form mRNA. Transcription is the copying of genetic information from the double-stranded DNA to form RNA, a single-stranded molecule, and does not contain a complementary strand. The RNA and newly formed complement of the antisense strand of original double-stranded DNA is the same as the one DNA strands, except for the replacement of thymine with uracil. The complement strand of the original antisense strand or RNA is formed using RNA polymerase, a similar process to DNA replication. This produces mRNA or messenger RNA, which carries messages from the nucleus to the cytoplasm. RNA polymerase moves in a 5' to 3' direction. It is important to understand that splicing, a process involving spliceosomes, exons, and introns, increases the number of proteins that an organism can produce. Translation, as described earlier, is the process of protein synthesis, in which genetic information which has been encoded into mRNA via transcription is translated into a sequence of amino acids in a polypeptide chain. Translation. Messenger RNA is fed into ribosomes. Ribosomes are a mixture of protein and the second kind of RNA, called ribosomal rRNA. This ribosomal rRNA does not contribute genetic information, but does have binding sites. These binding sites allow for the interaction of mRNA as produced from transcription to interact with another type of RNA, tRNA or transfer RNA. A ribosome is composed of two halves, a large and a small subunit. During translation, these small and large subunits assemble together. The small subunits bind to the mRNA, and the large subunit has binding sites, or tRNAs, which also catalyzes peptide bonds between amino acids. tRNA translates nucleotides, or the language of a combination of nucleotides, to form amino acids which go on to form proteins. Translation, as shown on this animation, depends on complementary base pairing between two codons on mRNA and anticodons on tRNA, which are a triplet of 3 bases with 64 different codon combinations, and how the codons can translate for 20 amino acids. Different codons can translate for the same amino acid, therefore the genetic code is said to degenerate, or be degenerate. The order of the codons determines the amino acid sequence for a protein. The coding region always starts with a start codon, methionine, meaning the first amino acid in all polypeptides is methionine. This codon is AUG. The coding region of mRNA terminates with a stop codon. The stop codon does not add an amino acid, instead it causes the release of the polypeptide. There are 4 steps involved in the process of synthesis of the polypeptide: initiation, elongation, translocation, and termination. Ribosomes found in the cytoplasm of cells bind with mRNA at the methionine codon or start codon. First tRNA molecules are brought in. Elongation, which takes up the bulk of the translation, and it's the addition of amino acids by the formation of peptide bonds. Elongation is simply the process of a chain of amino acids becoming longer. Peptide bond is formed between the amino acids, making a polypeptide. This step involves codon recognition and peptide bond formation. Once the tRNA has been used, ribosomes release them and move along to the next codon. Then, RNA and amino acid are brought it and elongate the same polypeptide further. When a stop codon is reached, the polypeptide is released and the ribosome picks up a new strand of mRNA. After a stop codon is reached, translation is stopped. It is important to note that free ribosomes primarily synthesize proteins for use within the cells that they exist in. However, proteins produced in bound ribosomes by translation are exported out of the cell. Translation in prokaryotes and eukaryotes can be different, as they have a different structure. Because there is no nuclear membrane, translation can occur immediately after transcription. An important characteristic to note of the structure of protein is that there are 4 possible levels of the structure, but to be able to check what level a protein belongs to is determined by its amino acid sequence. There are 4 structures for protein levels, and they are as follows: primary structure, secondary structure, tertiary structure, and quaternary structure. The specifications of these 4 levels of protein structures are displayed on the image below.